Elle, and I'm going to be giving you a lecture on membranes and transport with a focus on the role of proteins in those topics. So we've already covered phospholipid bilayers, and you should know quite a bit at this point in time about the chemistry of phospholipids. So I'm just going to remind you that that's a phospholipid, and that they make up the majority of the structure of a membrane organized into something called a phospholipid bilayer, but that about 50% of the mass of a membrane is actually made out of proteins. There's some adjectives for describing those proteins. For example, an integral membrane protein spans the membrane. A peripheral membrane protein sits on one side of the membrane or the other. A protein that has like a kind of a hole running through it or a channel is called a protein channel, or sometimes called a pore. And also that many of the proteins in membranes have a little bit of carbohydrate attached to them. They're called glycoproteins. They're used for a few different functions, one of which is for cell-cell recognition. And also in membranes we see steroids, which belong to the lipid category, for example, cholesterol, as seen in animal membranes. And we see some lipids combined with carbohydrates called glycolipids that play a variety of roles in cells. So that's like basically how a membrane is composed. We've already discussed the criteria for considering when you're looking at a chemical species and you're asking the question, will that chemical passively diffuse across the phospholipid bilayer? And over here we've got a phospholipid bilayer stacked up, and we're seeing that there are two categories of molecules here that are able to diffuse across through simple passive diffusion, while others cannot. The three criteria that we look at are the size of the molecule, the polarity of the molecule, and whether or not the molecule is neutral. So starting here at the bottom, ions are not neutral, they're fully charged. So anything with a full charge cannot simply passively diffuse across the membrane. Medium to largish molecules, especially those that are polar like glucose and sucrose, also cannot passively diffuse across a membrane. Little polar molecules like water, urea, and glycerol, and also CO2, which is quite polar, they are able to passively diffuse across the phospholipid bilayer, as are small non-polar molecules like oxygen and nitrogen. They can passively diffuse across membranes. Now there are lots of chemicals that cells need that cannot passively diffuse across. So for example, glucose, sucrose, or any of these ions. And so there are ways of getting them across, and those ways always involve proteins. And that's what we're going to focus on now. I'm going to skip over this part. We've covered it before. So, and as I said before, there's lots of terminology. Membrane proteins can be peripheral or they can be integrated. But while we're looking at this cartoon, I want to remind you that proteins are made out of amino acids. There are about 23 commonly occurring amino acids, at least in our own cells. And those amino acids can be polar or non-polar. And so when you're looking at a protein that's in the membrane, the amino acids are there for a reason, and so they're going to help to orient these proteins within the membrane. So you're going to tend to see sections of the protein that have non-polar amino acids and sections that have polar amino acids, and that's going to help a protein orient itself in the membrane. Because of that, we can use this term that we've covered previously in class. That term is amphipathic, meaning a molecule that has dual nature. So proteins can be amphipathic. They can have amino acids that are polar and even charged, and they can, and therefore hydrophilic, and they can have amino acids that are hydrophobic. And altogether, those amino acids, when attached to one another through the covalent bonds called peptide bonds, can form a larger structure, which is the protein, and that larger structure will have a particular function. In the case of membrane proteins, that function is going to require that the protein integrates into the membrane and stays in a particular conformation, so that it's not just like flopping around here in the membrane, but it's seated within the membrane and it keeps the correct orientation to do its job. For example, transport. So when we're talking about membrane transport by proteins, there are three sort of categories of the proteins involved: channels, which are often called pore proteins, carriers, and pumps. There are also three mechanisms of protein-mediated cross-membrane transport: facilitated diffusion using channel proteins, facilitated diffusion using carrier proteins, and active transport, which generally requires a pump at least somewhere in the process, but may also use a carrier protein. Facilitated diffusion with channel proteins means you have a chemical that is moving through a channel or pore protein, and that chemical is moving down its concentration gradient. So it's facilitated in that it uses protein, but it's diffusion in that the chemical is moving down its concentration gradient from an area of high concentration to an area of low concentration. So here's a cytoplasmic membrane, and here's a pore or channel protein. It's called aquaporin, and it allows water molecules to move through it down a concentration gradient. You know, water can also move across a membrane by simple passive diffusion, but it doesn't move fast enough to meet the cell's needs generally. And so it will also move through channel proteins. Facilitated diffusion that involves channel proteins can sometimes be reliant on proteins that are what are called gated. And gated means controlled. So in the example of aquaporin that we were just looking at a minute ago, that's not a gated protein. It's just open for business all the time. Always going to let water move. And there'll be a net movement if there's a concentration gradient. In contrast, here we have a protein that I have maybe mentioned before in class and we'll be learning about more in the future. It's a protein called ATP synthase. And this is a really ancient, really important and cool protein. One of its jobs is to make ATP by combining a phosphate group with ADP, ADP being adenosine diphosphate, and AT being adenosine triphosphate. And so this protein's job is to synthesize or build ATP. That's one of its jobs. And one of its other jobs is to transport protons. So protons will accumulate in the intermembrane space of the mitochondria due to the action of something called, well, anyway, it's part of aerobic respiration, and we'll be covering that later, so I won't get into it right now. It creates an accumulation of protons on one side of the membrane. They cannot passively diffuse across, but they can move through channel proteins that are intended for protons, such as the channels found in ATP synthase. And that movement of protons will power the synthesis of ATP. So here we're really focusing on the proton transport, and it is happening as a result of the concentration gradient, and that concentration is a gradient of both chemicals and charge. So if you have a lot of H pluses on one side of the membrane and not very many on the other, then one side will be very positive and the other side will be relatively negative. And that difference is called an electrochemical gradient. And it's so important in biology that it has a special name. We call it the proton motive force. And the proton motive force then powers the synthesis of ATP. Here I want to point out that ATP synthase is functioning as a channel protein to allow facilitated diffusion of protons, and that protein is only open for business when there's a certain electrical potential across the membrane. So that's called being voltage gated. Let's see. Another example of facilitated diffusion with channel proteins is cystic fibrosis, transmembrane regulator, or CFTR protein, which we all have. This is a protein that moves chloride ions, and it is controlled not by voltage, but by the binding of ATP to an inner part of the protein itself. On the left, we have a normal CFTR that's functioning to move chloride ions down a concentration gradient, here moving out of the cell. If that CFTR protein is absent or not working properly, we don't properly move chloride ions out of the cell. And as a result, the mucus in our respiratory system becomes very thick and sticky, and that is the condition known as cystic fibrosis. And facilitated diffusion can involve channel proteins, but it can also involve carrier proteins, which are shaped just a little bit differently. This is a protein called GLUT1. And it moves glucose down a concentration gradient. These little reddish molecules are glucose molecules. They will bind to the GLUT1 at a binding site. That triggers a change in the shape of the GLUT1. It then flips and it drops a glucose off on the inside of the membrane. And so all of this is driven by a concentration gradient, and it involves this protein GLUT1. Simple passive diffusion and facilitated diffusion both rely on chemical gradients or electrochemical gradients as the source of energy for transport. They don't directly use ATP. In contrast, most active transport uses ATP hydrolysis, which I'll show you in a minute, and often involves an electrochemical gradient created as a result. It generally involves moving some chemical against a concentration gradient. In other words, from low to high. So it's like pushing it uphill, pushing it into the higher concentrated area or side of the membrane. This is a way where we can, or a system whereby we can move ions and other larger chemicals, is to use active transport. Just so that you know what I mean, ATP hydrolysis means taking an adenosine triphosphate and with the help of a water molecule, breaking off an inorganic phosphate or PI, leaving ADP as a result. The converse or the opposite process is the synthesis of ATP, which I mentioned once before. ATP hydrolysis releases energy. It's an exergonic reaction. Active transport generally involves doing this, breaking down or hydrolyzing ATP. An electrochemical gradient is a term used to refer to an accumulation of charged ions where there are more on one side of a membrane than the other, creating a chemical and a charge gradient. Here is the proton motive force, a concentration of H+ or protons on one side of a membrane and not the other. So we have a lot of H+, a lot of the hydrogen ions, that's the chemical, and a lot of the plus charge associated with them, that's the electro part. And this is called the proton motive force. It's an example of an electrochemical gradient. Active transport can be primary or secondary. In primary, we transport ions across the membrane against their electrochemical gradient, for example, the proton motive force, and we use ATP to do this. In secondary, we use the electrochemical gradient, like the proton or motive force or another one, to move something else across a membrane. This doesn't directly use ATP, but it relies on the electrochemical gradient made by primary active transport. I just want to do a word of caution here, though, and I just don't want to mislead you. So I've written that primary active transport means to transport ions across the membrane against their electrochemical gradient. For example, the creation of the PMF. And the PMF is an example of an electrochemical gradient, but it does not use ATP itself to create it. Instead, the proton motive force is through the combined action of a set of proteins and organic molecules called the electron transport chain, shown here in blue. And it's powered by reduced molecules that come from earlier stages of aerobic respiration. So I just don't want to mislead you there. There's no ATP directly consumed in order to create the proton motive force. I'm just giving you the proton motive force as an example of an electrochemical gradient. All right, and with that, I'm actually going to close this so this video doesn't get too long.